Okay, so thank you very much for the introduction. What I thought I would try and do, since there's been some tweeting already, is ask a number of questions. My Twitter handle is 10 questions. There are 10 questions during this talk, which are to provoke you thinking, and during the talk, if you want to follow on with those questions, you can do that. So, the title is Bringing Viruses in from the Cold. Where am I from? First question. And how did I get here? You've been given a little hint in the introduction. Where is this? What is this? The dark edges, which are in Game of Thrones. So where am I from? Where am I from? Yeah, which bit? So I'm from Northern Ireland, which is where these dark edges are. That's where I'm from. You should be able to guess that from the accent. How did I get here? Well, that's an easier question, like everyone else, I think. So I've been in Boston for about 4 years now. I'm in the National Emerging Infectious Diseases Laboratories. This is a building in South Boston. Why did I come? Because this is a new facility which will allow us to work with dangerous pathogens. That is the title, that is the idea that I want to communicate with you this evening, working with dangerous pathogens, why we want to do it. Okay, how did we first see viruses? Well, we didn't have an electron microscope in ancient Egypt. How we first saw viruses was by seeing what they did. And this is a great example in a hieroglyph from ancient Egypt of a temple priest carrying a staff and his leg is withered. Which disease? Anyone guess? No? Which? Nope. Poliovirus, very good. This is the first recorded example of what a virus is. We saw the viruses by the diseases that they caused. We saw paralytic poliovirus in the United States because of massive infections in the middle part of the last century, where the virus attacked muscles and attacked lungs, and people ended up in these breathing devices, these iron lungs. How do we tame these viruses which cause these horrible diseases? Well, people like Albert Sabin managed to bring this dangerous pathogen which was causing this disease in from the wild, and he passaged that virus and he changed that virus, and eventually, if you're as old as me, you got little polio drops on your tongue, tasted very bitter, and that protected you from this very, very ancient disease. Vaccines are licensed. These people who were in iron lungs, not good for this guy, but for his siblings and his family, they could be vaccinated because people in the United States made a massive contribution to the development of vaccines to treat these horrible diseases. This is another example of a mummy, this time from Egypt, and this is a child with a disease. Which disease? Smallpox. Smallpox. Global eradication of a virus is possible by the use of a highly efficacious vaccine. So it was possible to remove smallpox from the world, and there's nowhere in the world apart from two places where you have vials of smallpox, in the United States and in the former USSR. In fact, the very good vaccine that has been developed by Sabin and Salk is leading to the eradication of poliovirus, and that has been slated for the next number of years. In your lifetime, you should see this virus eradicated. Okay, so what are viruses? Well, if we think of this as 1 cubic millimeter, what do they do? Here's a millimeter cubed, and in the corner of this millimeter cubed, I'm going to show you a very small red blood cell. And in between the red blood cell and the corner of the cube, this gives you an idea of how tiny, how minuscule these little agents, harbingers of disease, are. And because they're so simple, they have to build their structures by these repeating units, these little copies and copies and copies, that they can build these beautiful icosahedral structures with gorgeous symmetry that you see in this animation. Here's the subunit that the virus uses to build its shell. How many viruses are there? Well, I'm interested in viruses, and because there are more viruses in the world than the combined fungi, plants, animals, insects, and bacteria. Why is that? Because, as the nursery rhyme says, big fleas have little fleas upon their backs to bite them, and little fleas have lesser fleas, and so ad infinitum. One human, many viruses. One bacterium, many viruses. One plant, many viruses. So there are many, many, many different viruses. Here's an example of just the start of the A's. You don't need to read this, but you can see here the wonderfully termed angelfish reovirus. In fact, funnily enough, angelfish. And then you've got down here aphid lethal paralysis virus, which infects these little green fly. Where do viruses come from? This is important because we want to understand the origins of these viruses. And this is an important idea in biology, that animals act as reservoirs for infectious diseases. So here's a chicken and here's a lady. What disease from this chicken to this lady? Potentially. Yep. Very good. Avian influenza. What about this monkey? HIV is a good example. AIDS is the condition, HIV is the virus. And we're working on understanding the cross-species infections, which will stop animal viruses jumping into people. That's what we do in my research group. Why understanding transmission is very important is because viruses spread in these little droplets. And influenza virus spreads highly, highly efficiently. So this is a front cover from a newspaper in Brookline at the time of the 1918 pandemic, when influenza led to the death of 50 million people worldwide. What about bringing a virus in from the cold? That's the title of my talk. How can we get that virus, which is long gone in 1918? Well, in Alaska, there are a number of large grave sites where they're in the tundra, so this is the permafrost, and in the permafrost, these people who were succumbed to the infection had been essentially frozen, and what scientists were able to do was bring the virus in from the cold from those people, and they brought it in from the cold, and using the power of synthetic biology and virology, they were able to resurrect that virus and look at why that virus was so pathogenic. So, in the world of synthetic biology, is another question, eradication a redundant concept? Of course it is, because even though we can eradicate the virus, synthetic biology allows us to build it again de novo. Emerging infectious diseases. This is very popular. This is very, very current. There are many, many emerging infectious diseases. It's the National Institute for Emerging Infectious Diseases. I don't have the time to talk about all of these. I'm just going to talk about two. I've already mentioned influenza. We'll talk about Ebola. The other speakers are going to talk about Ebola later on. So I don't want to focus exclusively on Ebola, as interesting as that virus is. But we know that viruses can jump species. We know that avian influenza circulates in Asia. We know that bird flu can jump from birds to people and end up in hospitalization of children and adults. How do we study this? Why do we study this? Should we study this? Cross-species jumps of influenza. Well, someone recently did a very interesting experiment where they took a bird virus, and that avian influenza, they adapted it to a mammal. We're mammals. This guy here's a mammal. Ferret model of influenza. And as the New Yorker stated, he asked the question, did a scientist put millions of lives at risk? Was he right to generate the deadliest virus? A virus which is highly pathogenic in chickens, does not naturally infect people or mammals. He adapted it to mammals. And that has caused a significant amount of controversy. Papers were redacted, were not allowed to be published. There was a moratorium on the research. This is the guy who did the work here, speaking in DC. And scientists were blamed as irresponsible because they were adapting, making novel viruses with novel properties. Recently, 6 weeks ago, the United States has halted the funding for this type of research yet again. There's a new moratorium on these types of studies while the risks and benefits of those studies are determined. I had the pleasure of last time speaking not to you, a group of people like you, but down in the National Scientific Advisory Board for Biosecurity, where I was asked to defend these studies, to explain why we should generate viruses with enhanced transmissibility in mammals, why we need to work with dangerous pathogens. So we've talked about the history of viruses, we've talked about what they are, the diseases they cause, we've talked about cross-species jumps, viruses which jump from animal reservoirs into people. What about Ebola, this ongoing epidemic? Anyone know the potential reservoir for Ebola virus? What the idea is that where that virus is circulating normally? Shout out.
In bats.
Absolutely. Very interesting. Many people are looking at bats, sampling bats, looking to see where those viruses are acting as reservoirs, not killing the bats, but whenever they jump species from the bat to the person, they lead to significant levels of mortality in those cross-species jumps. The current situation in Western Africa is incredibly challenging. It is very fluid and it is desperately difficult to manage this outbreak. But one of the things which is most certain is protecting us demands protecting the people in Africa, controlling that outbreak at the source. So the ongoing epidemic is really large. This is where it is taking place in Western Africa. There's a significant number of cases concentrated in Sierra Leone and Liberia. This is the cumulative curve at the moment. Without going into the numbers, it's a massive outbreak. Over nearly 5,500 individuals have died from that infection in Western Africa. And that makes what's going on here with our small number of cases pale into insignificance. So we have to be careful to focus on where the problem actually is. How do viruses get here? I've told you how I got here. How did Ebola get to America? Well, a gentleman flew from Monrovia to Brussels, from Brussels to Washington, from Washington to Dallas. Pretty insignificant. Lots of people do that all the time. There are many flights. How did the virus get here? Well, of course, viruses know no borders. They can travel very efficiently in the incubator that is a person who's infected. So this unfortunate gentleman, the first person in America to die from that disease in Dallas, and actually managed to pass on the infection to two healthcare workers who were caring for him. This doctor in New York was more fortunate. You can see again, the press has a lot to answer for and how it covers some of these stories. I'm sure we'll hear about that, why the press needs to be sensible in how it covers these stories. Because our job as scientists is to inform. A good, sensible press should be there to inform, not to scare people to death, which is what sometimes happens. So, the problem with this wild, unsubstantiated speculation is we have to remember in the here and now, we are not living in a Hollywood movie like Contagion. We are living in a normal country which has a good healthcare system. And one of the questions which people have been asking, and I mean it's an unusual question, well, could Ebola change to become airborne? A virus which is not airborne at all, could it change? I was asked that question whenever the Washington Post phoned up in September to speculate on how many people might die. And the only way to answer that question is to say there is no scientific evidence. We have to be very careful not to scare people. Asking the question how many people might die in America of Ebola virus is the wrong way to look at this global problem. So, wow, why do we need to work on dangerous pathogens? Well, I believe in keeping your friends close and keeping your enemies closer. And these dangerous pathogens are our enemies. And the beauty of you working and studying in this university, in BU, is this is a university which has access to a state-of-the-art facility. The NEIDL is like nothing else. It is a wonderful facility where we can work with these pathogens in safety. And as much as I love my work, and as much as I love my viruses, the viruses that I work with, I don't want to go into this building to die. I want to go into this building to work safely and carefully with these viruses. This is the final thing I want you to, I can't bring you all into the building, it would be wonderful to do that. But we were very fortunate. This is a great science communicator, Vincent Racaniello, and TWIV, this week in virology. If you're interested in viruses, this is a wonderful podcast. You should listen to this. What the American Society for Virology did is they brought a camera crew into the NEIDL. They walked through it. We show everything, how we keep it safe, how we keep it secure, how we work with these dangerous pathogens. And you get a little look around something which your university has access to, and your university should be incredibly proud of having such a facility. So, why should we bring viruses in from the cold? I hope I've shown you what a virus is. I hope I've shown you what people do with them. I hope I've shown you a little bit about what happens whenever you heat them up and why you have to be careful when that happens. And I hope I've provoked you to ask a few more questions about virology and viruses. Thank you very much. So I will repeat the question, and hopefully I do it justice. The question is, what do I think about people who either knowingly or unknowingly come into the United States with Ebola virus? Well, one of the things that I think about this country, I'm constantly, not being from here, as I've told you and as you get, is that America is a tremendously developmental, generous nation. And I was saddened to see some of the reaction in the press whenever humanitarian people like nurses and doctors were brought back in planes that your taxes and your mother's and father's taxes have paid for, and brought them into the country, and the people said, you know what, they took the chance, they went to Africa to help these people, they got to let them die there. That's ghastly. I actually find that ghastly, and that for me is so not American. That's not the spirit of this country at all. So I think in that instance we should be careful, but we should realize that the problem is not here. The major problem is in Africa, and doing what we can to control that outbreak over there, that's the most important thing. Someone unknowingly coming back, I think we have to be careful. So we do have to have measures in place, but if you think of the nurse, someone who had tested twice negative for Ebola was asked to go into quarantine. It simply doesn't make sense, and this is where a scientist has to say, look at the scientific evidence. And in that instance, there was no evidence that that lady should not have been able to go. I think she also has to be responsible, because not everyone knows the viruses the way she knows it. And people do, like your mom, people do ask genuine questions and are concerned. But I do think that your government would not be welcoming people into the country who were a massive liability for this country. And I think we have to have a generous spirit in dealing with people who have gone to help others. I don't know if that answers the question. It does, okay.
Where is the facility that is in the picture?
So the facility in the picture is on the medical campus. If you know Albany Street, so whenever you're driving down Mass Ave, there's a big BUMC bridge. It's a building which goes across Mass Ave. If you go underneath that and turn left, that's Albany Street. There's a flower market and there's a prison on the opposite side, and there's the 93. It's in that general area. So one of the problems with the facility is a number of people have been very concerned about opening a facility which is going to work with Ebola in the middle of Boston, and that's caused BU a lot of problems. And actually, this is where communication, we were chatting about this just already, this is where communication is really important, and making the arguments, and simple things I think, like me saying I don't want to go into work to die, simply have no desire to bring an infectious disease in close proximity with me that has killed 5,500 people. But again, it's like back to your mom, we have this, we know this, and we can think about all of the safety and all of the redundancy and all of the containment. We also have a responsibility to tell our neighbours, our community, the people around us, why we think that this is safe, why we believe that we can work on dangerous pathogens, why we have to, and why whenever we bring Ebola into Boston, it can be managed in a facility using spacesuits, using state-of-the-art technology, that we can work with it effectively and really try to understand that pathogen better. Thank you very much for your time.